Have you ever wondered why plants grow healthy in fertile soil? Or how animals like us get the protein we need to build muscles and tissues? The answer to that question takes us into one of nature's most fascinating and invisible processes, the nitrogen cycle. Nitrogen is everywhere around us. In fact, if you look up at the sky and take a deep breath right now, almost 78% of the air you just inhaled is nitrogen gas. It's all around us, in the air, in the soil, and even inside our bodies. Yet, there's an interesting twist. Neither animals nor plants can use nitrogen directly from the air. That might sound surprising. We are surrounded by nitrogen, but we can't make any use of it in its natural gaseous form because it is chemically very stable. The two nitrogen atoms are held together by a strong triple bond, which makes the molecule almost impossible for most living organisms to break apart. So even though nitrogen is abundant, it remains locked away, unavailable to most forms of life. But life on Earth cannot exist without nitrogen. Every living cell depends on nitrogen, because it is an essential part of proteins, enzymes, nucleic acids, and ATP, the molecules that make life possible. So, if we can't use nitrogen gas directly, how do we get it? How do plants and animals obtain this life-sustaining element? That's where the nitrogen cycle comes in, a beautifully coordinated natural system that quietly works behind the scenes every day. It's like Earth's own recycling program, where nitrogen keeps moving from the air to the soil, into plants and animals, and back to the atmosphere again. The real heroes of this cycle are microorganisms, tiny, unseen workers in the soil and water who perform chemical magic. They take the nitrogen gas from the air and convert it into forms that living things can use, such as ammonia, nitrates, and nitrites. Through different chemical processes, these microbes keep nitrogen moving in a continuous loop that sustains life. Every time a plant grows, a leaf falls, or an animal excretes, some part of the nitrogen cycle is happening. It's one of those natural processes that connect all living and non-living parts of our planet into one interdependent system. So, the nitrogen cycle is not just a topic in microbiology. It's a story of how life recycles its most important building block. It means that the same atoms of nitrogen that once existed in old plants or animals might now be part of the proteins in your own body. Isn't it interesting? Let's try to understand how this happens. The nitrogen cycle completes in 5 steps: nitrogen fixation, nitrification, nitrogen assimilation, ammonification, and denitrification. Let's see the first step of the nitrogen cycle, the nitrogen fixation. Nitrogen fixation is the process by which atmospheric nitrogen gas is converted into ammonia or related compounds that can be used by all living organisms. There are 3 ways in which nitrogen gets fixed: biological, physical, and industrial nitrogen fixation. As the name suggests, living microorganisms like bacteria are responsible for the biological nitrogen fixation. The strong triple bond in nitrogen requires a large amount of energy to break. These bacteria possess a special enzyme called nitrogenase. The nitrogenase enzyme can break the triple bond of nitrogen and reduce nitrogen gas to ammonia. Nitrogenase is extremely sensitive to oxygen, and it stops working if exposed to it. So, these bacteria have developed clever ways to protect the enzyme while carrying out nitrogen fixation. The chemical reaction of nitrogen fixation can be written in something like this. These microorganisms capture nitrogen gas from the atmosphere and convert it into ammonia by adding hydrogen ions and electrons with the help of ATP energy. Once ammonia is produced in the soil, it can react with water to form ammonium ions, which plants can absorb and use. So, in the soil, most nitrogen is present as ammonium ions rather than free ammonia. The biological nitrogen fixation is one of the most important biochemical processes for life on Earth. So far, we have seen how the atmospheric nitrogen is converted to ammonia. Now, let's see what kind of microorganisms are involved in this process. Two types of bacteria are involved in biological nitrogen fixation: free-living bacteria and symbiotic bacteria. Free-living bacteria, or non-symbiotic bacteria, live independently in the soil and do not depend on plants for survival. They fix nitrogen on their own, using the energy from their metabolism. These are some of the examples of free-living nitrogen-fixing bacteria, which include both aerobic and anaerobic bacteria. These free-living bacteria take in atmospheric nitrogen through diffusion. Inside their cells, the nitrogenase enzyme uses electrons and ATP to convert nitrogen into ammonia. The bacteria then use it to build their amino acids and release the excess ammonia into the soil. When these bacteria die or release metabolites, plants benefit from the nitrogen compounds they leave behind. As we discussed earlier, the nitrogenase enzyme is sensitive to oxygen. For this reason, the aerobic bacteria like Azotobacter produce slime layers of polysaccharides to slow oxygen diffusion. Some cyanobacteria form special thick-walled cells called heterocysts, where nitrogen fixation takes place in oxygen-free conditions. The symbiotic nitrogen-fixing bacteria live in partnership with plants and form a mutually beneficial relationship, also called symbiosis. They mainly associate with leguminous plants such as peas, beans, clover, and lentils, but some also live with non-leguminous trees like alnus or casuarina. Examples of symbiotic nitrogen-fixing bacteria include Rhizobium, found in root nodules of legumes, Frankia, found in root nodules of certain non-leguminous plants, and Anabaena in Azala, a water fern. Under the limited nitrogen conditions, the roots of legume plants release chemical signals called flavonoids into the soil. Bacteria like Rhizobium sense these signals and respond by producing nod factors, which cause the plant's root hairs to coil around the bacteria. The bacteria enter the root through infection threads and travel deeper into the root tissue. An infection thread is a tube-like structure that grows from the plant's root hair, acting as a pathway for the bacteria to travel inside the root. The plant responds by forming small swellings called root nodules, which serve as tiny nitrogen-fixing factories. The bacteria inside the plant cells undergo morphological and physiological changes to form bacteroids. This process is controlled by the host plant. Bacteroids are capable of fixing nitrogen actively. The enzyme nitrogenase inside these bacteroids converts nitrogen to ammonia using ATP energy from the plant. The nodules contain a red-colored pigment called leg hemoglobin, which acts like hemoglobin in human blood. It binds oxygen and maintains a low oxygen environment inside the nodule so that nitrogenase can function efficiently. The plant quickly converts the ammonia produced into ammonium and then into amino acids, proteins, and other organic nitrogen compounds that the plant can use for growth. And the bacteria get carbohydrates and protection from the plant. This is how the plant and the bacteria mutually benefit from each other, creating a symbiotic relationship. When the plant dies or sheds roots, the fixed nitrogen is released into the soil, and the released nitrogen will be available for other plants. This is the reason why farmers often grow leguminous crops like beans or pulses before cereals, to naturally restore soil fertility. The physical nitrogen fixation is another method of nitrogen fixation that occurs naturally. We already know the air around us is filled with about 78% nitrogen gas. But this nitrogen gas is highly stable. It has a very strong triple bond between the two nitrogen atoms. However, nature has its own power tools, and one of them is lightning. The lightning power of nature converts the atmospheric nitrogen to nitrates through a series of chemical reactions. Nitrates are a usable form of nitrogen for the plants. During a thunderstorm, when lightning flashes across the sky, It produces tremendous energy and heat. The heat is sometimes more than 30,000 degrees Celsius. This high energy is powerful enough to break the strong triple bonds of nitrogen gas in the atmosphere. Once those nitrogen molecules split apart, they quickly combine with oxygen present in the air to form nitric oxide. This nitric oxide then reacts further with oxygen to form nitrogen dioxide. Now, when rain falls, the nitrogen dioxide dissolves in rainwater and forms nitric acid. This acid reacts with other minerals or bases present in the soil to form nitrates and nitrites, the usable forms of nitrogen for plants. Plants then absorb these nitrates and build proteins, DNA, and other vital molecules. In simple terms, whenever lightning strikes, it helps fix a small amount of nitrogen naturally into a usable form for plants. That's why the soil becomes more fertile after a thunderstorm, because rain brings down these natural nitrates to the soil. So far, we have seen the two methods of nitrogen fixation that occur naturally. Now, let us learn how nitrogen is converted to usable forms industrially. In nature, microorganisms use enzymes to convert atmospheric nitrogen gas into ammonia. But scientists found a way to achieve the same result on a much larger scale in an industry. And that's what we call industrial nitrogen fixation. The most important industrial method is known as the Haber-Bosch process, named after the two scientists, Fritz Haber and Carl Bosch, who developed it in the early 1900s. This process changed the world because it made it possible to produce fertilizers in massive quantities, supporting agriculture and feeding billions of people. So, how does this process work? In simple terms, the Haber-Bosch process combines nitrogen gas from the air with hydrogen gas to produce ammonia. Unlike bacteria, which use enzymes, we need very high temperatures and pressure to break the strong triple bond between the two nitrogen atoms. This process is carried out at around 400 to 500 degrees Celsius temperature and 200 to 300 atmospheres of pressure. A finely powdered iron catalyst, often mixed with promoters like potassium and aluminum oxide, is used to speed up the reaction. The hydrogen gas used in this process usually comes from natural gas like methane or other hydrocarbons through a process called steam reforming. On the other hand, nitrogen is obtained directly from the air, which is abundant in nature. As this reaction is reversible, the ammonia can again split into nitrogen and hydrogen. To avoid this, the ammonia is separated, cooled, and collected in liquid form. This industrially produced ammonia serves as the starting material for many important fertilizers, such as urea, ammonium nitrate, and ammonium sulfate, which are essential for modern agriculture. After nitrogen has been fixed into ammonia or ammonium ions, not all plants can use them directly. Moreover, high concentrations of ammonium are toxic to some plants. This is where the nitrification step comes in. Nitrification is a biological oxidation process in which ammonia or ammonium ions are converted into nitrite and then into nitrate by the action of specific aerobic chemoautotrophic bacteria. It occurs in two steps. In the first step, ammonia or ammonium is converted into nitrite. This process is carried out mainly by Nitrosomonas, Nitrococcus, and Nitrosospira bacteria. These bacteria use this oxidation reaction to obtain energy for their survival, which makes them chemoautotrophic bacteria because they use chemicals instead of sunlight as their energy source. Now, the nitrite formed in the first step is still not stable and can be harmful in large amounts. So, in the second step, the nitrite is converted into nitrate. This process is carried out mainly by Nitrobacter, Nitrococcus, and Nitrospira. By the end of nitrification, ammonia or ammonium is transformed into nitrate, which can be easily absorbed by plant roots and used to make proteins, nucleic acids, and chlorophyll. So far, we have seen the conversion of atmospheric nitrogen to ammonia and nitrates, which are the most usable forms of nitrogen. After the nitrogen is converted into ammonia and nitrates, through various processes like fixation and nitrification, what purpose do they serve if plants and animals don't utilize them? The whole purpose of converting atmospheric nitrogen into usable forms is that living organisms can use these compounds to build essential biological molecules. This is where assimilation comes into play. Assimilation is the process by which plants and microorganisms absorb inorganic nitrogen compounds, mainly nitrates and ammonium from the soil, and convert them into organic forms such as amino acids, proteins, nucleic acids, and chlorophyll. Plants usually take nitrate from the soil through their roots. Inside plant cells, nitrate is first reduced to nitrite, and then further reduced to ammonium through enzymatic reactions. This ammonium is then incorporated into amino acids through pathways like the glutamate and glutamine cycle, which are the main nitrogen donors for other amino acids and proteins. However, animals cannot directly absorb nitrate or ammonia from the soil. Instead, they obtain nitrogen by eating plants. The nitrogen present in plant proteins is digested and incorporated into animal proteins. So, while plants assimilate inorganic nitrogen from the soil, animals assimilate organic nitrogen by consuming plants. The assimilation process allows nitrogen to enter living organisms. Therefore, it ensures that the nitrogen does not remain unused in the environment. It is essential to synthesize all nitrogen-containing biomolecules, including proteins, DNA, RNA, and chlorophyll. It forms the foundation of the food chain, as all other organisms depend on plant-assimilated nitrogen. We have just seen how plants and animals use nitrogen compounds through assimilation. Eventually, the life of plants and animals comes to an end. When a plant or animal dies, its body doesn't just vanish. The dead plants and animals have plenty of organic matter in the form of proteins, DNA, and other nitrogen-containing molecules. Even while living, animals release nitrogen-based waste products like urea and uric acid in droppings. Now, imagine if all dead animals and their excretions keep piling up, and the nitrogen inside them is locked forever. Slowly, the soil would run out of usable nitrogen. Plants wouldn't get the nutrients they need to grow. Animals, in turn, would suffer because they depend on those plants. But nature doesn't let that happen. Instead, it has designed a brilliant recycling system. Microorganisms in the soil break down these remains and wastes, and release nitrogen back into the soil in the form of ammonia, making it available again for plants. This process is called ammonification. Ammonification is the process by which organic nitrogen found in proteins, nucleic acids, and urea is converted into ammonia or ammonium ions by decomposer microorganisms, mainly bacteria and fungi. These microbes break down the nitrogen-rich organic matter in dead plants, animals, and their wastes, releasing ammonia as a byproduct. Microbes first break down or decompose complex proteins into simpler amino acids. Then, deaminase enzymes remove the amino group from the amino acids, releasing ammonia. This step is called deamination. Once ammonia is released into the soil, it can react with water molecules to form ammonium ions. If the organic matter contains urea from animal waste, the urease enzyme breaks it down like this. And then, ammonia will be converted to ammonium ions. Ammonification is very important because it recycles nitrogen from dead organic matter back into the soil. The ammonium ions produced can either be used directly by plants as a nitrogen source, or converted into nitrites and nitrates again through nitrification, continuing the nitrogen cycle. It ensures that nitrogen is never wasted. It keeps moving through ecosystems. Now that we've seen how nitrogen moves from the atmosphere to living organisms through nitrogen fixation, and then back into the soil through ammonification and nitrification, There's still one final question left. How does nitrogen return to the atmosphere? That's where denitrification comes in. After plants and microorganisms have converted nitrogen into nitrates and nitrites, some nitrogen remains unused in the soil. If it kept building up, the soil chemistry would change, and nitrogen would get stuck in the ecosystem instead of circulating. So, certain bacteria convert these nitrates and nitrites back into gaseous nitrogen, which escapes into the atmosphere. This process completes the nitrogen cycle. Denitrification is the microbial process by which nitrates and nitrites are reduced to gaseous nitrogen forms under anaerobic conditions. It is mainly carried out by denitrifying bacteria, such as Pseudomonas, Paracoccus, Thiobacillus denitrificans, Bacillus species, etc. In waterlogged soils or deep sediments, where oxygen is rare, these bacteria use nitrate as an alternative electron acceptor instead of oxygen during respiration. Let's look at the step-by-step chemical reactions of the denitrification process. First, the nitrate will be reduced to nitrite in the presence of nitrate reductase enzyme. Next, nitrite will be further reduced to nitric oxide in the presence of nitrite reductase enzyme. Then, nitric oxide will be reduced to nitrous oxide in the presence of nitric oxide reductase enzyme. Finally, nitrous oxide will be reduced to nitrogen gas in the presence of nitrous oxide reductase enzyme. The overall reaction can be written like this. After nitrogen has been used and transformed many times in the soil, denitrifying bacteria send it back to the atmosphere as nitrogen gas. With denitrification, the nitrogen completes its cycle. Let's take a quick look at the overall nitrogen cycle.